This doesn't make any difference, right? I'll just check. You please sit. I'll check from another device. आवाज आ रही है? आवाज आ रही है क्या अभी? आ रही है अभी, रुक इसके बिना देखते हैं। अभी आ रही? अभी आ रही? It's working. So yes, so we'll have a very quick overview about this biotechnology and its applications, right? So we were talking about biotechnology principles and processes, that how you can actually modify the organism, right? This section basically is going to deal with the applications of the biotechnology, right? We know that biotechnology was basically dealing with genetic engineering. This live session will be divided into two sections, where I'll be giving a brief overview of this introductory part, which is added in the new NCERT. In the old NCERT, this was there in strategies for enhancement in food production, which is gone now. So in the new NCERT, you have all this. So I thought that we'll have overview of this. Then we'll have a brief overview of what are the basic concepts in this chapter, right? And then we'll be talking about the basic examples. So I'll take a break of 10-15 minutes. In the morning it was 1 hour live and I got like tired. So I know, matlab, sometimes I feel ki if I am getting tired, though, what about the students who are studying physics, chemistry, mathematics, biology, you know? I really pity you. But anyways, leaving aside that, so biotechnology and its applications, as I told, so it is like about the industrial scale. See, problems at the industrial scale when you are producing something on a large scale is more. And as I told, this is a revision session, so I'll be focusing only on the thrust areas. I won't be going into the conceptual details. maybe which will come in the we'll be discussing in the next sessions, right? So here we'll be talking about the industrial scale production of biopharmaceutical products and various biologicals, right, which are generally based on using genetically modified organisms, the organisms whose genetic makeup you have changed, the plants, the microbes, the fungi, the animals, right, in whom you have changed their genes. So using those organisms when you're producing something on a large scale, that is what is called as biotechnological applications. So where have we found these applications? We have found them in therapeutics, in treating the disease, in diagnosing and knowing the disease, in producing the genetically modified crops where you could produce or increase the yield, in formation of processed food, in bioremediation, that is cleaning up of your environment using microbes and all that stuff, that is bioremediation, in the waste treatment, and in energy production. So all these are the areas where we have used this biotechnology. So you have to remember that where we have used therapeutics, diagnostic, GM crops, processed food, bioremediation, waste treatment, and energy production. Three thrust and critical areas where we have got this more application is your identifying a best catalyst, which could be a microorganism or an enzyme which will produce your product of interest. Then optimizing the conditions, right, that is bioprocess engineering them so that the catalyst or that organism will give the maximum yield. So first I identify the organism, or I engineer that organism or that enzyme, and now I'm finding out the conditions which will work best for that enzyme. Matlab, at what temperature, at what pH, under which conditions the yield will be maximum. And then we'll be talking about the downstreaming, right? That is purification of that product. So how I'm going to purify my product from there, right? So purification of that protein or the organic compounds, right? The backbone of our economy as India, we have what is called as agriculture. So biotechnology has found wide application in this agriculture. How it is like based on using agrochemicals, or going for organic agriculture, or going for what is this genetically modified crop based agriculture. This is the thrust area. We have been using these agrochemicals and all that stuff since long time. In green revolution 1950s and all, we saw that there was a increased yield in food. We saw a record increase in wheat and rice, right? And that was basically because of two things. One, we used an improved crop variety, and second, we used the agrochemicals. We started adding fertilizers, we started adding pesticides, right? So these were the two things. You have an improved variety and you started using these agrochemicals, and you could actually triple the food supply. But then you will find that it created more issues. Like there was pollution problem. The fertilizers which were added, they were totally chemical. So they were like in the soil, they made the soil saline. The pesticides were totally chemical. So they were present in the food chains, and they were causing harm to all unnecessary, not unnecessary, all useful insects and even plants and animals. So we came up with a new branch, that is what is called as tissue culture, right? And although these chemicals, the fertilizers, the pesticides, the chemicals which are there, they are quite expensive. So we came up with another idea of what is called as tissue culture. So tissue culture basically is growing your plant in a controlled laboratory conditions. That is what is called as tissue culture in short. So what I do is I generate a whole plant from the explant. I bring a leaf from the plant, and I make the entire plant from that leaf. I bring a root, I bring a stem, even I bring one cell. And I bring that one cell in the class, oh, not in the class here. I bring that one cell in the laboratory, provide it proper conditions, and grow the entire plant. That is what is called as tissue culture. In the sterile condition, providing it nutrient media, obviously. In a condition where bacteria is not growing, fungi is not growing, so that all the nutrients can be utilized by that plant cell. And I'll provide it a proper nutrient medium. See, plant is not now growing outside. It's in your laboratory. So it is now your responsibility. It is your baby there. So you are going to give it a proper temperature, a proper pH, a proper food to that plant so that cell it grows, right? So the ability of one cell or that explant to give rise to an entire new plant is called as totipotency. We don't have that, unfortunately. Or fortunately, we are fortunate enough we don't have that. Otherwise, we would have made so many of ourselves only. So anyways, so totipotency, as I told, the ability or capacity to form the whole plant from that explant or that plant cell is called as what? Totipotency, right? when you are providing a nutrient medium, what care you have to take? They are going to give it a carbon source, but it's not photosynthesizing there. It is not from or coming up from a seed which will have a stored food. So you're going to provide it with sucrose, you're providing it with salts, with vitamins, with amino acids, with various growth regulators like auxins, cytokinins. So you're actually trying to mimic all the natural environment which the plant must have got when it was in the soil or it was coming up from a seed. So nutrient medium has to contain all this: a carbon source, amino acid, vitamin, growth regulators, salts, right? Auxins, cytokinins in a proper amount, and then it will grow. Now, when I grow many plants, right? Like I have a rack, and I have a rack like this, and I'm growing plants on this. Like if it's in a small bottle, I'm growing a plant, and like there are 10 plants here and 10 plants like this. So I have 100 plants in this one rack, and I'm having 20 such sections. So I'll have like 100 into 22,000. And such I am having 10, it will be 20,000, and such I am having 5, it will be 1 lakh in one room. So growing many plants in short duration, that is called as micro propagation. Usko hum kya bolte hain? Micro propagation. When I am growing many plants in a short duration, that is micro propagation. And all these plants are going to be exactly same. So they are called as soma clones. We have heard about this word called as clone. So all these plants are exactly same. So they are called as soma clones. So here are the examples you have to remember is tomato, banana, and apple. So the questions which might be asked. So here you may have a question like which plants have been micro-propagated and produce soma clones of. So it is your tomato, banana, apple. You may have a fourth option, potato. Though you may love potato and vada pav, but it's not that, right? It is your, just a second. Muskan, please call mommy. So here you have this tomato, then you have banana, and then you have apple. So these are what you call as produced by micropropagation and these soma clones, right? So you can also get a healthy plant from a diseased plant. Many plants, they get infected with the virus, and now if you are taking any explant from there, or if you're even propagating that plant through the seed, you'll find that the plant still has a disease. If you take the apical meristem, it may be apical or it may be axillary. The meristem is generally free from the virus. So if I take that meristem, which is free from the virus, and I'm now able to produce the entire plant from that one cell. Now that one cell doesn't have a virus, so this entire plant also doesn't have a virus, right? So I'll produce this healthy plant from a diseased plant. So I can, for that, I'll use a meristem, which may be axillary or apically, and it is free from the virus. So here you have to again remember that we have produced these virus-free plants in banana, in sugarcane, and in potato. So here the examples are banana, which is common in both the cases. There it was tomato, and it is batata, tomato and potato. There it was apple, here it is sugarcane. So virus-free plants, the examples, banana, sugarcane, and potato. Yes, here you have the potato. Maybe they heard that when I said like that. So when you isolate a single cell and you digest the cell wall, sometimes very difficult to digest some people's words, but here you are digesting the cell wall, right? But then you take the single cell and then you digest the cell wall, and what you get is the protoplast. And now you take that naked protoplast, which is surrounded by the plasma membrane, and you take these two protoplasts and fuse them. And what you get is a somatic hybrid. This is what we have been trying to do when we are doing a traditional biotechnology. So what we were trying to do is we were trying to produce hybrid plants. So you did a lot of stuff. What you did is that you took pollen grains from here and took androecium from here, and you mixed in, and then you got, here it's very easy. You take one potato cell, you take one tomato cell, remove their cell walls, mix it, now you have pomato, right? So you have a potato and tomato combined together to form pomato. Unfortunately, it doesn't have all those characters which could be like used in a commercial production. So it's not a very successful one. If it had been successful, you would have only one vegetable out there. So if you want to make batata chutney and tomato chutney, you would have been only one fruit or one that vegetable, not fruit actually. So, but tomato is a fruit, but potato is a stem. But we are not doing morphology. And tomato as exile presentation. Oh, we are not doing morphology. So potato and tomato, as I said. So potato and tomato gives me pomato, but is not showing all the characters. So that mixing is called a somatic hybridization. The process is somatic hybridization, and that mixed stuff which I get, that is somatic hybrid. So see why we are studying here, that this is possible only because of tissue culture. अगर टिश्यू कल्चर नहीं होता तो आप मिक्स किए हुए सेल्स का क्या करते? आप भाजी थोड़ी बनाते. आप क्या करेंगे, मिक्स किए हुए सेल्स को अभी टिश्यू कल्चर से उसको टोटिपोटेंसी की वजह से यू कैन excuse me, develop an entire plant. So that is the essence of tissue culture, right? So we can genetically alter the plant, the bacteria, the fungi, the animals, and then you call them as GMO, that is your genetically modified organisms. When I'm genetically modifying plants, I should have some aim to modify that. You know, like you are watching this video, you have some aim, like you want to crack NEET. So like when you are deciding to genetically modify a plant, you should know what you're going to modify, what is your aim. So the basic aims for which we are modifying it, number one is to make them tolerant towards abiotic stress. So if there is a lot of heat, there is a lot of cold, there is no water, there is a lot of salt. So against cold, drought, salt and heat, that is abiotic stresses which the plant is going through. To reduce the dependence on chemical pesticide, this is going to be the immediate point of discussion. If you're going to make that plant resistant to that chemical pesticide, so then you will have a pest resistant plant. Or to reduce the post harvest loss, like if you somehow you could what you call as make this like tomatoes when you are growing. So when you harvest the tomatoes, they become like they ripen, and once they ripen, they become soft, they lose their market value. If somehow you could retain their shelf life, if they could remain green for a longer period of time, then the post harvest loss rate would be reduced, right? So you can go for that, you can reduce the post harvest loss. Or you can what you call as increase the nutritional value. Like the very famous is that golden rice. So you're not going to take the rice and put it in the gold, but then you're going to put the gene of vitamin A. So now that rice, when you cook the rice, so in advertisement they show कि चावल नहीं बने, चावल बदलने से बहुत कुछ बदल गया. तो यहाँ पे चावल बदलने से बहुत कुछ क्या बदल गया, न्यूट्रिशन बदल गया, क्योंकि वो चावल में आपने क्या डाला है, जीन विच इज कोडिंग फॉर विटामिन ए. You will increase the efficiency of mineral usage, so that the plant, it efficiently uses the mineral, it doesn't waste it, so it retains those nutrients in the soil. And alternatively, you can use them for excess production of starch, for production of fuel, and for production of various pharmaceutical products. So these are the targets. You see, we can go for abiotic stress, for chemical pesticide, for reduction of post harvest loss, for efficient mineral usage, for increasing the nutritional value, and there you have to remember this golden rice, that is vitamin A. So this is what, like, the first section it deals with, and then the next section it basically starts with more specific applications of this biotechnology. Like here we saw so many applications. If you see one by one, what I'll do is, before going off and again coming live with a lot of stuff and all lot of information stuck on this board, like let us go with the conceptual understanding of what things they have told. Like first is the concept of what is called as Bt cotton. So we know that there is a bacteria called as Bacillus thuringiensis. You remember we saw this in the earlier chapter. We also saw this in microbes in human welfare. And this Bacillus thuringiensis is a bacterium which is what you call as having a gene which is called as the cry gene. And it is this cry gene which makes the insects cry. So various lepidopterans, dipterans, coleopterans. I'll be going with the specific example in the next shot there. But here you just remember that this produces cry gene, which produces the cry protein, which produces a toxin which is in the inactive form, and it becomes active only when it goes into the alkaline, what you call as conditions of the gut of the insect. So this toxin is in the inactive form and it goes and into the intestine of that insect and becomes active, creates holes in the intestine of that insect, and the insect cries and dies. If I could just clone this gene, that is cry gene, into the plant, now the plant itself becomes insect resistant. You're getting my point? So you can use the different cry 1AB, cry 2AB, cry 1C. In the next shot, I'll do that. So again, your cotton bollworms, again your corn borer. So you can specifically use that gene, and you can use that gene and insert into the plant, and you can make the plant resistant to that. right? So that you can do if you are using this Bacillus thuringiensis. So Bacillus thuringiensis gene you can use to make the plant resistant towards insect. Another thing which they have talked about is making it resistant to a nematode. And here you are going to use the phenomenon of RNA interference. Cutting it short, like if you are talking about a tobacco plant, so a tobacco plant gets infected by this insect Melodygaene incognitia, a nematode which is going to infect this tobacco plant and going to destroy that. We use the phenomenon of RNAi, that is RNA interference. See, I tell you, RNA interference is a phenomenon wherein what is going to happen is, if there is a cell and I'm having an RNA, and if I'm having a double-stranded RNA complementary to the same RNA, then this double-stranded RNA somehow, I'm not going into technical details, maybe we'll meet some other day after your NEET, but then somehow this double-stranded RNA, it destroys or cleaves this actual RNA and it stops its expression. So if I want to stop the expression of a particular RNA, what I'll do is I'll introduce a double-stranded RNA which is complementary to this RNA which I want to stop. That is what is called as RNA interference. And this is a natural mechanism by which plants are protecting themselves from various viruses which are having RNA as the genetic material, from various transposomes which use RNA as an intermediate. I know this is getting more complicated, but then now, as I told, the exams are near and we are going to focus only what is given in the book. So what you have to remember is, if I am having a double-stranded RNA and it is complementary to the RNA, this double-stranded RNA presence will cleave that RNA so that that RNA won't be expressed. Now what I'll do is, I'll have a RNA specific to this worm, that is Melodygaene incognitia, and I will take this RNA as a starting point, the gene of this RNA now, which is coding for that sense and antisense both. So I'll take this sense and antisense gene and I'll put it into the plant cell. Now the plant cell is producing the double-stranded RNA, right? The worm, it comes and it ingests this double-stranded RNA goes into this worm. Now in the worm cell now, this double-stranded RNA, it creates a phenomenon of RNA interference. And the important RNA of that worm is destroyed, is cleaved. And that is very important in the feeding process of that nematode. And then once that RNA is destroyed, the worm is also destroyed. So nematode is destroyed. So a plant is protected. So the RNA interference is used in controlling of these nematodes. So point to remember without much thing is that you have a double-stranded RNA specific to the worm, which is having both sense and antisense genes inserted into the plant. It goes into the worm and destroys the RNA of the worm. And once the mRNA of the worm is destroyed, the gene is silenced. The worm cannot survive, the nematode cannot survive. And who is protected? Your plant. Maybe after listening to this, you are asking for someone to protect you, but now I cannot do anything. So you have this Bt cotton, then you have this nematode. Unfortunately, this is only conceptual. Now the board is going to get filled with the information. But anyways, I hope you're getting the concept right here. Another basic thing of this biotechnology is that we are producing various hormones, like you have insulin, you must have heard of, produced in for patients who are suffering from diabetes. So this insulin is a hormone which reduces the blood glucose, right? It is produced before biotechnology. Now with biotechnology, a bacteria is producing this insulin for you all. But before biotechnology, it was being bought from slaughtered cattle, slaughtered pigs. Very ridiculous, you see. Killing the pigs, taking out their pancreas and extracting the insulin, and then you taking that insulin. Then who will eat those pigs? Sorry. I mean, like, how much you're going to get? Na? So much of waste is being generated, and ultimately it is foreign. So it is going to cause harm to you also. The problem with this natural insulin was that in human, when this is being produced, it is produced as a polypeptide which contains A polypeptide, which contains a B polypeptide, which is now joined with a C polypeptide. So it is synthesized as a prohormone. So it is synthesized as a prohormone which has 3 sections, A, B and C. And in nature, what happens is this C1 is cleaved and this A and B are joined by disulfide linkage. So processing takes place. So you have a proinsulin which is inactive, and this inactive insulin is now what you call as processed. The C peptide is removed, and then you join the A and B, and then you get active insulin. Now the problem was that how will you get this active insulin in a bacterial cell? It was in 1983. 1 year just before I was born, they thought that we should make some discoveries in science. So in 1983, there was a company which I really love, Eli Lilly, right? So in 1983, this Eli Lilly came, and what they did is they took a separate gene for coding for segment A. They took a separate gene for coding for segment B, put it in two separate E. coli cells. See, now I have a gene which is coding for segment A. I have a gene which is coding for segment B. I have put them in separate E. coli cell. I grow them separately, I produce them separately, and then I mix them. Then I'm going to mix A and B, and artificial now I'll create the disulfide bond, and I'll produce this active insulin, which is saving now millions of people around the globe from this dreaded disease, that is diabetes. So that is one way it is done. Another area where biotechnology is playing a major role, as I told, I'm just giving a brief overview of the idea, the basic concept I'm giving, the crux of the chapter. I'm worried how the board will be filled after this. Right. Anyways, so another thing which we can do is what is called as a gene therapy. So nice to imagine that your entire genes, it could be changed. So gene therapy is like you can change the gene of the organism. So you take the organism and you could actually replace the gene. A gene is not working. A gene is irreparable. A gene is like mutated. And therefore, the person is suffering from the disease. Like here also say insulin is not there. So what we are doing, we are producing insulin. And now you are injecting that insulin. But it's not going to remain there forever. You have to continuously keep on taking that insulin. A person who is suffering from diabetes has to keep on taking that insulin injections for a lifetime. But think of a situation where you could actually make that person make its own insulin by changing its genes. So that can be done with gene therapy. If you could, just a second. Yeah. So if you could actually change it, as I told, right? If you could change the genes, right? If you would replace the gene, that is what is called as gene therapy. If I am replacing it in any of these cells, then it is somatic cell therapy. But if I am replacing it in the germ cells, like if I am replacing in the sperm, if I am replacing in the ovum, probably I am replacing it for a generation, from one generation to another. So that can be done with this gene therapy. The first gene therapy, they have told, it was done in around 1990 with a 4-year-old girl which was having a deficiency of the enzyme called as ADA, adenosine deaminase, an enzyme which is very important for proper functioning of our immune system. So if you could like take this particular gene, ADA gene, and you could put it into that girl's lymphocytes, and she is now producing a good lymphocyte. So that can be done. You can also go for bone marrow transplant. But then from where you will get the donor and how it will work, we don't know whether it will match or not. Or again, if you are taking these lymphocytes and injecting those lymphocytes in them, then that also you will find that if lymphocytes are not immortal, they are going to die. So even if you are replacing it with good lymphocytes, but then again, this is not a very good thing to do, because what will happen is lymphocytes will die, again you will have to inject that. The good idea would be that if you could go at the embryonic stage and you could replace the gene of this ADA, then now the problem is solved from the root. And we always say that समस्येला मुळापासून काढून टाकायचं. So that मुळापासून काढून टाकायचं, the मूळ of everything is the gene. If we could actually replace those defective genes, I think nothing can be much better than that. So we are still in the process of replacing all these genes at the embryonic level. Another way where this has found application is your diagnostics, you see, where we are trying to diagnose a particular disease. So how you are going to diagnose a disease, like now you go to a doctor, they will go for serum analysis, they will go for urine analysis. This is what is done. But you know that it takes time, it takes more sample. By the time probably there are some severe diseases where the patients can die, maybe they are not very accurate. So for diagnosis also you can go with PCR, you can go with recombinant DNA technology, or you can go with ELISA. So these are some modern techniques which can actually help you to diagnose a disease at a faster rate. Even in this COVID, you remember they were taking swabs, and the test which they were doing was this RT-PCR, the reverse transcriptase PCR. So it is a rapid test. The sample which you require is very less. So with a very less sample and a very accurate test, you can diagnose that particular disease, that is with your PCR or your RDT, the recombinant DNA technology. You can detect a genetic disorder also. You can have a probe which is specific for a disorder, and you can label it, and then you can check whether the patient has the complementary probe or not, which is complementary to that genetic disorder. So if it is not there, then the person is healthy. If it is there, then the person is suffering from the disease. ELISA, the enzyme linked immunosorbent assay. If you remember, this was the test which was used as a preliminary screening test for AIDS. It is a serological test. It is based on the antigen antibody interactions, right? If I am detecting an antigen, I should have an antibody for me to detect. If I am detecting an antibody, I should have an antigen. So ELISA, I'm not going to details of the test because it's not given in your book. It is just based on this antigen and antibody interactions, right? You go with this biotechnology and produce what are called as the transgenic animals. So you go for production of these transgenic animals, the animals in which all your genes, they are changed. They are called as transgenic animals. And we have produced many of such transgenic animals. We have produced sheep, we have produced mice, we have produced rats, frogs, and many more. 95% of them, they are mice. So most of the work, I don't know why we have done with the mice, but then we do. Because it's easy, their lifespan is less, and many more stuff, but you're not given. So most of the times what we generally go with is your transgenic animals is with the mice, right? For what purpose we do this? We are doing this for knowing the proper physiology and functioning. If you want to identify like what role is being played by a particular gene. You remember in Marathi there is a in Hindi also people say कि किसी के चले जाने के बाद आपको उनकी अहमियत पता चलती है. So here they have found out, like, if there is an insulin-like growth factor, you want to find out what is the role of that insulin-like growth factor. So you have another gene which is expressing something which will stop the expression of this insulin-like growth factor here. So this will not function. And now you will see that if this is not functioning, the insulin-like growth factor, what effect it is having. So now you know that what is the role of this insulin-like growth factor. So you can identify the normal physiology and functioning. You can identify the disease, many diseases which are genetically what you call as determined, the cystic fibrosis or your autoimmune disorders or your Alzheimer's disease. You can actually find out like where we can use, right? You can use these transgenic animals for vaccine safety, you know. People used to use those monkeys. So instead of using those monkeys to detect those polio vaccine safety, you can go for using these mice, right? So that can be one thing. You can use for as a what you call as production of these various biological products, as a, you can say, a factory for production of these chemicals. Like they have produced, you know, see, they have told that you produce that human milk enriched cow, you know, which was Rosie, and who is producing that alpha lactalbumin at a rate of, I think, around 2.4 grams per liter, right? Then they have produced various compounds like alpha-1 antitrypsin, which is used for treatment of this, which is that disease, cystic fibrosis, the alpha-1 antitrypsin, right? So different products, the biological products you can produce, right? So that can be done. Then you can use them as factories for that production. You can use for chemical safety and testing. What happens is that these are very sensitive. The transgenic mice, they are very sensitive. So if you have produced any chemical or something, and if you want to test whether it is toxic or not, you can test it on these. So if they are toxic to them, they are going to be not safe. But if they are not toxic to these most sensitive organisms also, probably they will not cause harm to the other sturdy organisms, right? Biotechnology, it also deals with various ethical issues, you see. It is not only about doing what you want to do. You also need to decide the ethics, that what is right and what is wrong. So when I am deciding the ethics, so you need to understand that these ethical issues, we have in India agency called as GAC, Genetic Engineering and Approval Committee. And this is this GAC which will decide that whether the new product which you have produced is actually good or not. So that GAC you'll find is the committee which will be approving your genetically modified crops or genetically modified plants, animals, like when you're going to put it in the ecosystem, whether they are going to cause any harm to the nature or whether they're going to harm to the ecosystem. You need to decide that. Another problem which comes up with these ethical issues is that you cannot just play with the human genome. It is not ethically, morally, spiritually right. People don't actually accept. Society doesn't accept. So, like I told, BT cotton as we started. We have BT rice, not BT rice, but I think there is BT brinjal, BT tomato and all. But then they are not widely accepted. Right here, these ethical issues, they need to be addressed. Right. So you see, there was an example of this rice. Right, rice, we have a very documented varieties. We have around 27 documented varieties of basmati rice, and in India itself we have around 2 lakh different varieties of rice. We have such a huge biodiversity. But then we don't patent all these. We don't have patents for all these. Patent is a right which is granted for the person who has invented that process or discovered that new plant or animal or microbe or anything, and then he can use that, right? So this particular rice also, you know, that there was a American trademark office which is there. They issued what you call as patent to these people regarding the use of this basmati. So they took this local growing basmati rice and mixed it with some semi-dwarf varieties, and now they produced a new variety like Texamati or something like that. And now there were restrictions on selling the same variety or using that same variety, which is not correct because it was our origin. So people in the developing countries, the problem is that they have a lot of traditional knowledge, they have a lot of resources, but they don't have a lot of money, and they don't have technical knowledge for patenting their products. Like we also have won a case against turmeric and neem. So people now have become vigilant, and now we are fighting for all this because this is our traditional knowledge. No one can take away that from us without giving us proper what you call as compensation for that. So if you are doing that, that is what you are doing is biopiracy, which has increased a lot these days. So there is a patent, right, which is the permission which is given to you. And then there is biopiracy when you are using others' resources and others' knowledge without giving a proper compensation to that individual, to that organization, or to that country at large. So that is not correct. There are laws against this, and you can be sued for that, right? And Indian government also is now what you call as very vigilant regarding that. And recently a bill has been passed regarding this particular concept of patenting and having the rights over our own traditional knowledge and traditional methods, right? I think instead of like filling the board like entire thing, we'll just read out like what is given in the text, and we'll see and remember those specific examples, right? So coming back to the first thing which we started, the first thing was that BT cotton one, right? In that there was this Bacillus thuringiensis, right? I'll just be back. So let me read it out for you. I know that you can read it on your own, but then let me help you out to remember some important points here. So when we are talking about this BT cotton, so it produces these insecticidal proteins. Here what you have to remember is those lepidopterans. Lepidopterans, they have mentioned two, right? The tobacco budworm, right? And they have mentioned this armyworm. So tobacco budworm and this armyworm, they are lepidopterans. Then coleopterans they have mentioned. And these coleopterans, they are your beetles. And then you have these dipterans. And dipterans, they are these flies and your mosquitoes. Right, the remaining concept I already explained, that you can take this gene and you can put it there, and the crystals, they become active. That is in the inactive form. So it is the protoxin. So the gene, it becomes active only in the alkaline conditions. And the gene which I had not told, that was your Cry1AC, that is called as the Cry gene. And the two things which I told, it is Cry1AC and Cry2AB, that works against cotton bollworm. And then what remains is your Cry2AB that works against corn borer. Right, so that you have to remember. So it is Cry1AC, Cry2AB, it is cotton bollworm, and Cry2AB, it is your corn borer. Pest resistance, I already talked, I think everything I told about Melody incognitia, infecting the roots, you have to remember, reducing the yield. You are using the phenomenon of RNA interference. You are having a complementary double-stranded RNA, which is stopping the translation. It is silencing of mRNA, protecting from viral genome and mobile elements of genes, that is your transposons. Some other day we will discuss about this transposon. And then agrobacterium, you have to remember. See, whenever I am transferring any gene to the plant, the basic vector which I use is the bacterium called as Agrobacterium tumefaciens. So that you can do. Here you have to remember, when you are transferring the gene, you have to transfer the gene of both sense and antisense. This is the point you have to remember, that you are going to transfer the gene for both sense as well as antisense, because you don't want a single-stranded RNA, you want a double-stranded RNA. And you're going to silence the gene of whom? The farmer, the tobacco, or that nematode? Whose gene you're going to silence? The farmer, the tobacco, or the nematode? Who want to kill in this process? The farmer, the tobacco, or the nematode? The nematode, right? So you're going to use that gene which is silencing that nematode gene. So transgenic plants are protecting itself from the parasite. Then we talked about in medicine, I think I already discussed about this, that you have those pro-insulin, which is in two forms. Like it has pro-insulin, which is having a C peptide. It becomes active after you remove the C peptide. And RNA technology came in. Major problem with this was that it could not be assembled. That was the major problem. This was solved in 1983 by Eli Lilly. They were growing it in the same bacteria or different bacteria. Different. A in different bacteria, B in different bacteria. Which was the bacteria used? E. coli. Yeah, so that could be extracted and then combined by these disulfide bond. Then we talked about gene therapy, where you could change the gene. First, I told 1990, a 4-year-old ADA deficiency, and that was the first step. You can use the lymphocytes. Here, what I forgot to mention is that to transfer that gene, you can use a retroviral vector, a retrovirus. So that can be used for transferring that. Lymphocytes, I mentioned that this question can be asked, that why lymphocytes can be transferred, then why are you going for gene therapy? So lymphocytes can be transferred, but lymphocytes are not immortal. So you have to transfer it again and again. So to avoid that, you go for what you call as embryonic stage gene therapy. Remember, adult stage gene therapy and embryonic stage gene therapy. What is more efficient is the embryonic stage, because that will be a permanent cure. Then we talked about molecular diagnosis. If you remember, I told we diagnose disease by serum and urine analysis. I told this can be done by PCR, ELISA, and RDT. So pathogen you can detect by PCR. The value which you require, it will waste the amount of sample. You can just multiply. You can detect even the mutations. So that can be done. You can use the single-stranded radioactive molecule as the probe, and then you can detect whether this is present or not. So that will help you to identify the complementarity with that mutated gene. So here you have to remember, the clone having the mutated gene will not appear in the photographic plate, because the probe will not have complementarity with the mutated gene. So that you have to understand. Then ELISA, based on which principle I told? Antigen and antibody, that you have to understand. So detect the antibody synthesized against that antigen. Transgenic animals, I told, for normal physiology, for studying of the diseases, right? The cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. Fortunately, I mentioned all of them. Then biological products, I told you can make. What I forgot here was, I think, the treatment for phenylketonuria and cystic fibrosis. But we mentioned alpha-1 antitrypsin, which could be used for treatment of emphysema, right? Phenylketonuria for cystic fibrosis. And then 1997, the first cow, that was Rosie, 2.5, I told it was your alpha-lactalbumin. Vaccine safety we told, and chemical safety also we told. Here you have to remember, the question which may be asked is, transgenic mice are being used to replace these monkeys, which were earlier used to test these vaccines. Then we talked about ethical issues. I already told that is your GEAC, which is giving you approval for that, for publicizing or using it at a broader scale. In India, we told that there are how many varieties? 2 lakh varieties of rice, and how many documented varieties of basmati? 27. And I told that 1997, American company got this patent on basmati. US and abroad in this new variety, right? It claimed that other people could not sell basmati rice, right? And it was restricted by this patent. That was the major thing which happened. We have also problems with this turmeric and neem, which are our traditional, what you call as medicine, our legacy. Using them without proper authorization from the country or people, and without giving them a compensatory treatment, I told it payment, it was biopiracy. And then I told that developing countries have a lot of traditional knowledge, whereas developed countries have a lot of money. So they are exploiting those traditional. To stop that, you need legal actions. And I told Indian Parliament, here you have to remember that we have passed the second amendment of Indian Patents Bill. That you have to remember. And we are considering such issues so that any provisions related to these patents and all, they could be sorted out. This was a very quick overview of this chapter. It is very difficult because this chapter we generally teach for at least a week, and to complete this in around 40-45 minutes was a tough task, but with your help we did it, right? So that is about biotechnology. So stay tuned. Tomorrow morning again we'll be meeting, right, with some chapter. I am also not sure which chapter, but then now we are in a phase that on 5th we are having exam, so anything you studied is in the syllabus. Earlier what used to happen, like this is not within the syllabus, or this has been taught, or this revision is still pending. So we are just revising. When you get bored with the books, you can get bored with me, right? So thank you. That 3 was